So as Rayla and Paul Lambert already indicated, a lot of our speakers today are alumni of the Temin Lab, most of the speakers, in fact. And the next speaker is a good example. This is our next keynote speaker, John Coffin. And as most of you know, John did his PhD work with Howard at the old McCardle building, which is just north of us here across the street. And John was in the Temin lab when the existence of reverse transcriptase was so strikingly proven. But he left before the Nobel Prize was awarded for that work. And he left to do postdoctoral work with Charles Weissman in Zurich, University of Zurich, continuing his study of retroviruses. And then from Zurich, he moved to Tufts University, where he's a distinguished professor of molecular biology and microbiology. And at various times, he has augmented his contributions there with activities at other sites, NCI and elsewhere. John's many scientific accomplishments include numerous fundamental findings about retroviruses and their host interactions, and that includes, among other things, their genome structure, their reverse transcription mechanisms, mutation rates, integration patterns, responses to treatment, and other properties, as well as his studies on endogenous retroviruses and other retroelements that are embedded in our genomes. And his recognitions include an American Cancer Society professorship and election to the National Academy of Sciences and many others. And I'm very happy to turn the floor over to him to hear the lessons that he has for us from these experiences. John.
Thank you, Paul, very much. I am somewhat at a loss as to how to follow Rayla. It was really wonderful to hear the story. She said a lot of things that even I didn't know, and I'm certainly the oldest alumnus of the Temin lab here. In fact, there was only one graduate student ahead of me, and he finished about 2 or 3 weeks before I did, so giving Howard the wonderful opportunity of having to edit 2 theses at the same time. In my talk, I will start out by talking a bit and covering some of the same ground, but from a somewhat different perspective than Rayla. I covered, of course, from this perspective of a graduate student in the lab. And I think I will add a few little hopefully interesting tidbits to here and there. And then I'll go on at the end and talk a little bit about what I've been doing since and what I consider to be my proudest achievement, actually. So when I joined Howard's lab in 1967, as a graduate student, as I've already heard, actually, 2 things were true. First, and this is what I heard this actually said in a course I took, what's true for E. coli is true for the elephant. And second, of course, is a central dogma, which my good friend and our colleague in Frederick likes to say, called a shaggy dogma. And that was Steve Hughes who coined that term. And in which the information from DNA could be passed to RNA, could be passed to protein, and those 2 could pass information to molecules of the same type, but only because it had never been seen. There was no chemical reason why this couldn't be true. The information could not be passed from RNA back to protein. Because it didn't happen or hadn't been seen to happen, it obviously couldn't happen, and that was the obvious logical flaw in the dogma. So, bring in Howard Temin. Howard got into the field actually working with a postdoc in the lab, Harry Rubin, And in 1958, he and Harry published a paper that really set the stage for everything, which, as Rayla went on, talked about, was on devising a focus assay for Rous sarcoma virus, and this was the first quantitative assay that allowed quantitative genetic experimentation on any retrovirus. And and one of the first things he did was to use it to look at the genetic property of RSV, as was already discussed by Rayla, the shape of the transformed cell, and this shows the actual pictures from the paper on that. And so let's see if I can work this pointer. There it is. Wow. Okay, so here are round cells by one strain of RSV, and here are the fusiform cells that Rayla mentioned, the elongated sort of spindle-shaped cells. And these two were isolates that were originally derived from the same original isolate of RSV, so they're obviously had to be in some way closely related. And so this is the actual statement from the discussion of that paper. There are two ways in which RSV could operate. Either it could contribute genetic information directly to the cell to enable it to become tumorous, or it could activate a tumorous state of the cell. And he was very cautious in the discussion of this paper, but it was not hard to read through the lines to see exactly what he was thinking at the time. He was, of course, unable to draw the conclusion that he later did draw about the nature of the provirus and so on that you've already heard about. So Harry Rubin, however, was having none of this. Harry was a very different kind of scientist from Howard. He's a good scientist. He had a very good career. He trained many well-known retrovirologists. But he was a believer more in methodological, one-step-at-a-time kind of science. He thought Howard's intuitive leap into thinking that there was some genetic change that the virus could cause in a cell in some specific way was just too far out. And this led to actually a rather famous feud between them for at least the next decade. I think they eventually made up, but many, many years later. And that actually became quite well known what was going on there. And Howard went on, as Rayla again mentioned, to publish a number of papers. Here's three of them that she also mentioned, actually, the actinomycin D paper, the 1964 paper implying DNA, and a paper that I'll show you a figure from in which labeled RSV RNA, just grown in the presence of tritiated uridine, was hybridized to DNA from RSV-infected cells or control cells, and he reported out the amount of RNA that had annealed to this DNA that you can see in this paper. However, this paper did not draw much acclaim, I would say. It was not widely believed. For one thing, the amount of activity that was seen in the positive case was very small. It was only 3 counts per minute, a tiny, tiny amount. And there was also, it was only about twofold above background. In retrospect, it's quite likely that background was real, and it was something that he wouldn't have imagined at the time. Even Howard didn't imagine at the time that there was already some virus genomes in the cells, endogenous virus that he was working on. And those actually became a major subject of my research much later on. Finally, in 1970, Satoshi Mizutani, who was a relatively new postdoc in the lab, found the killer experiment, which was likely motivated by David Baldimore's work on RNA polymerases in virions, which he had shown actually just the previous year, and reported. David and Howard reported some preliminary experiments on this at the same Gordon conference, and they were probably cross-motivated each other to actually do the final experiments. At least that's the way I view this. And the experiment was a very simple one. You take a preparation of concentrated virus, you break open the virus and you add some labeled preparation of labeled nucleotides, and you then just measure the amount of high molecular weight material, which DNA that's made, and this shows the result. Now it's thousands of counts, not just 3, and it's a very simple experiment. And Howard presented, as Ray also mentioned, at the 10th International Cancer Congress. What I want to point out, this was in a session that was chaired, as it turned out, by Harry Rubin. And this was after 10 years of their sort of fighting about the provirus model. And Howard came back from this meeting, and I was there, I remember it very well, bubbling over with having really finally stuck it to Harry in a real good way. That he said that Harry, all Harry could say was, "I don't believe it, I don't believe it, I don't believe it." And and he asked around the lab whether he should really go ahead and publish the work right away, or whether he should wait and do a few more experiments. I advised him to do some more experiments, particularly to get the better characterization of the DNA product. That was the worst advice I ever gave. Fortunately, he ignored it. And a few days later, he in fact got a call from David Baltimore, who told him that David had done the same exact experiment and had already submitted his paper to Nature. Howard rushed and got the paper out to Nature in a very short time. This was already in late May, early June, and fortunately, Nature agreed to hold David's paper until Howard's appeared and could be published so they could be published together. According to a memoir written by Václav Sobalsky, it was Sobalsky that actually called John Maddox, the editor of Nature, to get him to hold the papers together. The result of this was, if you look at the dates, the paper was submitted, the envelope of the paper in it arrived at Nature, I believe it was June 15th, and the paper was published June 27th of the same year. In fact, it was actually published before that. The journals usually arrive a few days before the publication date on them. So, I believe a record for Nature. It beat out Watson and Crick. I did check that one. So, and the papers were very much the same. They were carrying out the same reactions. David's was actually done with murine leukemia virus. Howard's with, and Satoshi's with Ross sarcoma virus, as you've heard. The one apparent difference was that the David paper does not mention the use of detergent to open the virus. And they always attributed that to the fact that David used a virus preparation that he'd gotten from a federal contractor, and therefore was so beat up you didn't need to add detergent to it to break it up. I've never actually, David says that at least in subsequent experiments he always did use detergent, and he had used detergent in the experiments they had done with RNA viruses. But anyway, so this is where my first major contribution to the story came in, and that is that Satoshi borrowed my bottle of NP40 to do these experiments. I have another major contribution that I'll mention in a few minutes. So, the atmosphere around this, the scientific atmosphere, was, if you weren't there, it's something I've never seen before in my life. And there were many reasons for it that I'll get into at the end. But there was about monthly, there was a new thing on reporting the latest bit on reverse transcriptase in the news and views in the front pages of Nature. The first of these was actually from the Houston meeting, and Ray already showed a quote from that one. Then there's breathless news and views, central dogma reversed, and then just on and operate Temin le deluge unabated, advice for Teminizers. I think we saw that one already. More of the same. Now, this particular News and Views has another interesting aspect. I believe that this is the first in-print use of the term reverse transcriptase. The cell biology correspondent mentioned here was John Tooze, who's quite a well-known virologist as well. And he coined the reverse transcriptase, the term reverse transcriptase. He admitted it to me once when I challenged him with it. And this was written by him, and it's almost certainly the first time it was used. And so then, and after that, it's always reverse transcriptase. And then finally, a year later, we have the happy birthday. Okay, so, and also, Rayla's already showed you this. This led to lots of things, and I believe was also an important precipitating event on getting the war on cancer going. It also put new life into a special program, NIH program called the Special Virus Cancer Program that had far more money than ideas, and it breathed new life into that, and I think it sort of played a major role in re-energizing sort of molecular cancer research. So, since that time, I went on to be a postdoctoral fellow in the University of Zurich in the laboratory of Charles Weissman. This was actually at Howard's recommendation. Howard essentially set this up for me. So I told him we wanted to go to Europe because it was a thing that was done, very, very commonly done with American postdocs at the time. It became much, much less common these days. But and then after leaving there, I became a member of the faculty of Tufts University School of Medicine, the best medical school in the country for lunch, by the way. They're in the middle of Chinatown. And then in 1997, I took on a part-time job as a founding director of the HIV drug resistance program of the National Cancer Institute in Frederick, Maryland. I'll talk about that a little bit more later. Like many of the Temin alums, many of whom are here, I stuck with retroviruses for the rest of my career, focusing initially on RSV, then successively on MLV, various both exogenous and then eventually endogenous viruses, and then eventually human endogenous viruses, as well as issues of genome structure, replication, pathogenesis, evolution, and interestingly, nomenclature. And the nomenclature for HIV has already been mentioned. I was involved in that as well, and eventually turning my interest into HIV. So in the next few minutes, I'm just going to present a couple of vignettes, if you like, building on my training in retrovirology and scientific reasoning I received from Howard when I first learned, as now I tell my students, to think like a virus. So some of the first studies we did in now in Zurich, as I learned more about reverse transcription and the resulting DNA, it was pointed out, this was at a meeting that I was at by David Baltimore, that it was becoming ever more embarrassing to realize we didn't know how big the genome was of any retrovirus. And the reason for that is that, I'm sure as many of you know, retroviruses don't have a single molecule of RNA in the genome. They have, well, we now know two. It was 2 or 3 at the time. It was rather approximate. And we didn't know if those genomes molecules were identical in sequence or different in sequence. Now, viruses like influenza where are segmented like that, where the segments are all different in sequence and basically different genes, were already well known. But we did not know that for retroviruses. There were no known viruses at the time, in fact, still, as far as I know, that had identical molecules with multiple RNA, genomes with multiple RNA molecules that were actually the same as one another. Okay, so to answer this question, we used a 2D gel fingerprinting technology that had originally been devised by Walter Fiers that was being used by the Weissman lab as a tool to aid in the sequencing of a Q-beta bacteriophage RNA that they were interested in at the time, that most of the lab was interested in, actually. So this is one of those fingerprints. When I first saw one of these, I said, "There's the genome. There it is. There's my virus." And these are incredibly reproducible. You can do them at different times, different weeks, and so on, and lay the films on top of one another if you're a little careful about how you do it. And these are done by labeling the RNA uniformly with p32, so every nucleotide is labeled to the same extent, cutting with a ribonuclease, T1, which cuts only next to G residues. So what you get are a series of oligonucleotides, small products, some of which by chance will be quite large. Will be so large that there'll be nothing else that size or that base composition in the genome. And if you then run it out on one of these gels where you separate it by base composition, in this case by the number of U's, and then by length in the other dimension, you get these nice series of spots, many of which are, that's not what I want to do, that's what I want to do. So many of which are big enough that they would be only present once in the genome. And if you can determine the length of these and the proportion of the genome that's in there, the proportion of the counts, the amount of mass that's in them, you can very simple arithmetic, you can calculate how big the genome is. It doesn't matter how many pieces it's in. You can calculate whether it's all, all the pieces are the same or all of them are different. And so, based on this kind of analysis, it became very clear that the genome comprises now 2 identical subunits, and that had never been seen before. And we can talk a little bit, perhaps, about why this is. But another interesting feature is this little smudge you see next to spot 13 here. I should have ignored that. I ignored that at the time, but in fact, that proved to provides a very interesting clue to another interesting property of the genome that I will come to in a minute. So after I left Zurich and returned to Boston to the job I still have, basically. One of the things we got interested in was not much at the time was known about how reverse transcriptase actually happened. Sort of the fundamental biochemistry of the enzyme had been worked out, but how it actually copied the RNA, what were the actual steps in copying the RNA, nobody knew. They were slowly worked out by other labs. For example, it was determined that there was a tRNA molecule in the virion that served as a primer for DNA replication. And DNA synthesis of RSV and everything else we know pretty much requires a pre-existing primer to add on to. And in this case, the primer was tRNA. And it was discovered that the tRNA molecule, pointed out by John Taylor at Penn, that the tRNA molecule was somewhere near the 5' end of the genome, which is a rather odd place for it to be because the DNA synthesis would go in that direction and it would very soon hit the end of the genome. So when I first heard this, I thought, well, the only thing that could be was that it could be that there's a sequence here at the end of the genome that's also found at this end of the genome. That's the only way I figured this could work. And as it happened, Bill Hazeltine had been working on the DNA products, and the most common DNA product in the in vitro experiments that one does with this is a single molecule of about 101 nucleotides long called strong stop DNA. And that's because the methods to disrupt the virus also disrupted the organization of the genome that was necessary to go on further. And we knew also that there was a ribonuclease H associated with it. So so he had this molecule, and I had a method of seeing where this was coming from by using this fingerprinting technique. And so we got together. Actually, the first meeting that we had to get together was on the very day the Nobel Prizes were Howard and David were awarded. I happened to be in the Baltimore lab at exactly that time, by interesting coincidence. And so we were able to show, actually, that's exactly what happened, that we hypothesized, that this R sequence, and we made a copy of it here, and then the virus, that could be moved or jumped, as we like to say, over to here. And so this is the first step in reverse transcription. We did not work out any of the remaining steps. Those were left to others. And so you got this very really lovely scheme here, leading eventually to this molecule of DNA. This is now what becomes the provirus, which has these long terminal repeat sequences, although the RNA has just this little tiny repeat here. I did make one more interesting, I did publish one more interesting paper on a critical observation here, and that was what I take to be the shortest sequence ever published. Probably repeat, that honor is probably shared 1,000 times over, but the single nucleotide, the actual nucleotide at the site of, or the last nucleotide opposite the primer, in fact, matches the primer. It had previously been reported by others that it didn't, and that's a U residue. So this process was worked out for the younger ones entirely without any of the tools we have for modern molecular biology. It was worked out. There was no molecular cloning. There was no PCR. There was really no sequencing beyond single nucleotides at a time here and there. But the whole thing was worked out that way. And it's also identical for all retroviruses up to and including HIV, and for many retroelements. Also, the diploid genome, as I pointed out actually in reviews not about the same time, and the ability of the RT to jump from one molecule to another also explains the high rate of recombination in these viruses. Okay? And in fact, some of my colleagues who are here have gone on to work on this for quite a number of years afterwards, until quite recently, actually. So if we jump ahead 15 years, so over the years, Howard had been following my career, and I'm sure the career of everybody here, and everybody who was ever in his lab. And from time to time, he's sort of urging me to, maybe you should get into HIV. You should get in and start working on HIV. Well, I was doing pretty well with other viruses and endogenous viruses and other things at the time. So I kind of put that off for a long time. Until some experiments arrived that were some published experiments brought back to mind an experiment I had done when I first moved to Boston, and that was a classic experiment in virology, which is just to take the virus you have, passage it over and over and over again, and let the virus do whatever genetics it's going to do, whatever evolution it's going to do to the new conditions it finds itself in, and so on and so forth. And then look at the genome somehow to see what's happened. And so that was the experiment I did. I just passaged this virus over and over and over again, and took out samples and subject to exactly the same kind of fingerprint assay I showed you before. The property of those fingerprints is that the position of the spot there is a very sensitive indicator of the exact nucleotide sequence. So a single point mutation in that spot will move it. It might make it bigger or smaller, or it might just move it from one place to another. So you can measure the accumulation of mutations that are occurring in about 5% of the genome of your sample, and get a feeling for what kind of evolution occurs just under these simple passaging conditions. And so, to back up a little bit, up until that time, the shaggy dogma for HIV was that it was a lentivirus. Lenti is Latin for slow. So therefore, it's a slow virus. It replicates like other viruses, where there's maybe just a few generations from when one host is infected and the virus is transmitted onto another host. And that's because a virus like RSV doesn't kill cells. The cells that were originally infected are, in a sense, still around, or their progeny are still around years later when the virus can be transmitted again. But unlike other retroviruses, which show very little genetic diversity, HIV becomes very diverse in a fairly short time, even within a single individual, after clonal infection of a single individual. Therefore, the mutation rate, the underlying error rate of the reverse transcription process for HIV must be much, much higher than that of the simpler retroviruses. So, and then inconsistent with this, using the principles of neutral evolutionary theory, which assumes a small effective population size, that is, in my terms, the effective population size of the virus is much less than the inverse of the mutation rate. So the mutation rates are very, very rare. Mutation will only occur once, basically. So, so they use this with this assumption to conclude that the population size is small, without sort of thinking about the circularity of the logic here. The inevitable failure of current HIV monotherapy, the first was AZT, for example, which worked great for a couple of weeks, and then the virus, mutant virus would come up, was thought to be due to the appearance of resistance mutation, was due to the appearance of resistance mutation, but was thought that those arose as a consequence of incomplete suppression. You just provided a selective pressure. This is all about to change. And so this was the result of the experiment I showed you before, the passaging experiment, where you could see the kinetics with which mutations arose. And this is the result of a passaging experiment, actually of SIV, but closely related virus. The results would be exactly the same with HIV. And you see, when you passage them in cell culture like this, you really can't distinguish them. Okay? So even though if you isolate two different chickens from different parts of the world and get one of these viruses out, it's almost exactly the same as the one you're working with in the lab. But what this must mean is that's not a difference in underlying mutation rate, because that's what's driving this experiment. It's a difference in the basic host-virus interaction that is really causing the difference. And so what sort of triggered me here was a couple of papers that appeared from two different labs recently, showing that using the rate of decay of HIV after you start therapy in an infected individual, a person living with HIV, that the virus is actually turning over quite rapidly. That the infected cell only lasts about a day and a half before and dies, stops making virus, and then goes on to the next, which actually effectively gives you the replication cycle time. So in the course of the year, there are hundreds of cycles of replication of this virus, whereas there might be 2 or 3 or 4 in the case of a virus like Rustacoma virus. Okay. So, so I published this essay actually in Science in 1995. This is by far my most cited paper, and where I just point out that the mutation rate of HIV is not different from that of any other retrovirus. The effective population size is very large. Therefore, the replicating virus population, although usually clonal to begin with, will diversify to contain all possible mutations very soon, within a few weeks. The inevitable failure of anti-HIV monotherapy can be attributed not to low potency of the drug, not to sanctuary sites, but to pre-existence of the drug resistance mutations at the time you start therapy, because people don't start therapy with HIV until they're diagnosed, basically. And that's not for weeks and weeks, at a minimum, weeks and weeks after they're infected. By that time, the game is over as far as drug resistance mutations. That was the conclusions from all of this. Combination therapy, where you use 3 drugs at the same time with different targets, works not because it's more potent. It works because the frequency of these pre-existing mutations is individually quite low, and when you multiply them together, it's so low that there won't be a single genome in the whole replicating population that has all 3 of them. And so about the same time, I was approached by George Vanderwood and Steve Hughes and Harold Varmus to fill some space in a building that had been built to dedicate with HIV money and to dedicate to HIV research. And this became the HIV drug resistance program. It's building 535 on the NCI Frederick campus. It's now 3 floors of this building are now the home of the HIV DRP, now called the Dynamics and Replication Program, because drug resistance still remains quite a strong focus in this group, but it was thought to be a solved problem, although it really isn't, and it may well, there is at least one context in which it may come up to be very important, what's happening currently. So I was asked to recruit some individuals to start this program. And so the idea, of course, was to provide a program in which there's a big interacting group all focused on the same sorts of goals, but from very different kinds of science, from structural biology to all the way through to clinical biology, clinical virology. And so the DRP has been successful beyond my wildest dreams, I have to say. At one time I thought I'd work there for 5 years, part-time, and then sort of bow out, and I found myself unable to do that because it was so exciting, the work that was going on there. Not surprisingly, about half of the initial recruits were UW alumni, all of them with connections to Howard, either as being directly mentored by him or at least influenced by him at the time of their training. Although I was forced to step aside as director in 2005, I have still been pleased to be associated with that pretty much up to the present day, and still continue to be impressed by the productivity of this group, which you will hear from 3 of in subsequent talks. So, and just a long list, a few examples of these are on this slide. This list was actually borrowed from Eric Fried, who made it for another purpose, and it's only a partial list, actually. There are many more than this. So it was sensitive assays that became the gold standard laboratory tools for measuring virology in infected individuals, demonstrated the absence, so this was a big debate for a while, of detectable HIV replication evolution during suppressive therapy, discovered and characterized persistent viremia, discovered the HIV residue comprises, this is again a big surprise, expanded clones of cells infected prior to HIV, infected prior to ART initiation. This was a big surprise. It shouldn't have been. We should have known that. We should have figured that out ahead of time, but it wasn't, and we figured it out and published them. Deciphered the origins of the famous XMRV that was the virus that was not the cause, as we showed, of chronic fatigue syndrome, and Vinay and I collaborated on this one. And I will never forget, I'm sure he'll never forget the day we sat together in my office and put our sequences together to show exactly where the virus came from, which was from the recombination between 2 endogenous viruses in mice. They developed the use of HIV integration sites to study the reservoir, discovered capsids remain intact during reverse transcription, and many more. These are all things that actually are textbook stuff. Many of these have caused the rewriting, particularly the last one, has caused the rewriting of everybody's replication figure in every single textbook that was ever published up until that time. So, a couple of concluding thoughts. Retrovirus discovery had an enormous scientific impact. It was dramatic and compelling evidence for Temin's provirus model, and we heard about the derision that Howard was subjected to. He'd come back to the lab after a meeting very depressed, because they'd almost laughed at him. In fact, sometimes they actually did, I think. And so just the sheer drama of suddenly finding the killer experiment after all of these experiments that convinced him, but other people didn't quite believe and were unwilling to shed their shaggy dogma, was really a major scientific experiment. The key experiment was done by 2 labs at the same time and easily and frequently reproduced by others. In fact, there's a story that when David presented this at a meeting, it was in Cold Spring Harbor, Saul Spiegelman, who's at Columbia, heard the talk, drove back to his lab in New York, and returned the next morning to report that he repeated the result to the same meeting. It provided the key entree into fundamental basis of both retrovirus replication and their ability to cause cancer. It rapidly became as an important tool for modern molecular biology. In fact, I just looked this up. The market for the 2024 for commercial reverse transcriptase is over $300 million. It's a means of retrovirus discovery and assay, unfortunately rather often misused in this particular case, but it's still extremely valuable. For example, HIV was basically discovered this way. As a target for antiretroviral drugs to effectively treat AIDS and perhaps other retrovirus diseases if they appear. As an enabler of clinical gene therapy, which is obviously becoming more and more important, and marked a turning point in the basic molecular biology and its application to cancer. But it was also accompanied by an amazing amount of hype. There's been nothing like it, in my opinion, as far as hitting the press and all kinds of other things. It breathed new life into the cash-rich but idea-poor special virus cancer program, provided an impetus for Nixon's war on cancer. It was touted as providing a path to a cancer vaccine and cure, which has only been sort of indirectly true, but it certainly has been important, but obviously not in the direct way that was visualized here. But there's also a background of this anti-war zeitgeist that was going on, particularly here, right after the first meeting that I was at in 1970, where there was a lot of discussion of reverse transcriptase. I came back from this meeting, was walking from my apartment back to McCardle. I came by an area of the campus that was barricaded all off. I asked a policeman what was happening. He said, somebody didn't like your math building. So that was the day that Sterling Hall was bombed, killing a postdoc working late at night. And of course, about the same time was the shootings at Kent State. And there almost daily, there were crowds of students marching down University Avenue, being accompanied by clouds of tear gas, the smell of which sometimes drifted over into the lab. So it was quite a time. Then finally, the sheer drama of a lone scientist with a brilliant but heretical insight, supported only by rather indirect evidence, convincing no one, finally finding a simple and convincing proof after a decade of struggle against a wall of skepticism. And I'll close there and just show you a picture that I've carried with me now for 50-odd years of showing the Temin lab at the exact time. The exact composition of the lab was taken a couple months later at the time of the discovery of reverse transcriptase. So you can see Howard, of course, is here. Satoshi Mizutani is right next to him here. There were two other graduate students in the lab, Eddie Moore at the time, Eddie Moore and David Bediger. I'm hiding over here. Vicki Kastner, who's in the audience, is right here. And this, I believe, was taken this is Atsumi Kato here, was taken at the time, I believe, of her departure from the lab. That's why we were gathered together. If anybody wants a copy of this picture, I'll be happy to send it along. I scanned it. It was a little print. Was badly abused with the time of my scanning and moving it and putting it on the bulletin boards. I sent it to one of those sites you can download to clean it up, and it really came back beautiful compared to what it was. I could show you the original if you wanted to see it, but it was. Anyway, I'll close there, and thank you for your attention.
Okay, I think we've got time for one or two questions here. I just want to say that Rayla preferred to take questions at the break, but I think she would be prepared to take questions at the break if people are interested in doing that. So do we have any questions from John from the floor here? While we're looking for those, I'll just say I really am hoping that our students took, among other points, the lesson from your talk that many of these results that they think of as textbook bedrock were not in fact brought down from on high by divine revelation, but were were actually the fruit of hard, dedicated effort, and that they can remember this when they're facing their own challenges. So again, questions here? Yes.
I'm just going to make a comment. I was really struck, John, by the slide that you had where the effect was so small, and yet Howard went on. And I was reminded that that's often the case, that the initial discovery by Bob Lehman in Arthur Kornberg's lab of DNA incorporation was just a couple of counts above background. And that you just got to pursue those things if you've done the experiment correctly.
But yes, exactly. But it also, unfortunately, provides fodder for anybody that does not want to believe you, that thinks the handed down from on high, i.e., Watson and Crick, basically, and Francis Crick especially, shaggy dogma was in fact the real story. Oh yeah, sorry, you're absolutely right. That spot actually was due to the fact that spot 13 contains within it the entire R sequence. It was very short in RSV, it's much shorter than other viruses, and which means there was no G in the R sequence. So at the other end, you didn't see it because it ended up attached to the poly A. But occasionally, about 15% of the time, as we later showed, and I had some slides here, but I had to take them out, we later showed that about 15% of the RNA is read through. The polyadenylation that creates the 3' end of the RNA is not 100% efficient. Actually, the same thing was shown was rediscovered like many stuff many years later for HIV. And what we were seeing there was the spot at the other end with the first G afterwards, which comes from the U5 sequence. And if I had looked at that more carefully, the other thing was that the spot 13 actually contains the capping group. So if I had properly analyzed that, I could have discovered that too. In fact, we were also trying to get RNA out of infected cells. We were never able to prepare good RNA from infected cells for some reason. But if we had, we would have discovered that the lower molecular weight RNAs also contained that spot 13. And we would have perhaps discovered splicing as well. So you can see all of these wonderful opportunities to make all of these discoveries, fundamental discoveries, sort of pass me by ignoring that one spot. Yes. That's a nice way to say it. Thank you. I'll keep that in mind.
So
Really.
thank you, John, for filling in so much of the science and the background and with your incredible knowledge of the development of the whole field. So I left people in suspense about the vial.
Oh, you're right. I didn't mention that.
So I don't want them to go on to the
I ignored that. I didn't ignore it deliberately. I just sort of forgot. So with Howard looking up ahead at this tea flask filled with cell culture medium, I filled that flask.
Well, on that note, we'll move on here. Thank you so much, John.